Hello everyone, welcome back to my channel. So this is actually continuation of cerebellum. This is part 3. Many of you have been asking for this part 3 and because of the time constraints I couldn't do it. So those who haven't watched the previous 2 sessions, please do watch so that you will get a continuity for part 3. So cerebellum, the external features we have already covered. Now in this session I'm going to talk about the cerebellar cortex. So before we mention that, let's see how the cerebellum is arranged, the white matter and the grey matter of the cerebellum is arranged. So we know that the cerebellum is actually folded into many leaf-like folia. So that in the outer portion you have the grey matter concentrated as the cerebellar cortex, and the inner portion you have the white matter coming from this grey matter, and the innermost portion again you get some of the grey matter arranged as the intracerebellar nuclei. So you have the cerebellar cortex with grey matter, then you have the inner white matter, and the innermost portion you have the grey matter again arranged in the form of intracerebellar nuclei. So these are the folia, and between the folia you have the transverse fissures. So when you take each folia, you can see that there is a rim of grey matter which is the cerebellar cortex, and from the middle, from the innermost aspect, you have the white matter which is joining the white matter core. So let's see how the white matter is going into each folia. So if you just have a look at the white matter alone, you can imagine it as branches of a tree. Okay, so if you take a section of cerebellum, these white matter going into or coming from the cerebellum will be looking like branches of a tree, and hence that section, that appearance is known as arbor vitae cerebelli, or otherwise known as tree of life. So once again, in the cerebellum, the grey matter and white matter are arranged as if the grey matter forms a periphery known as the cerebellar cortex, and it is also seen within the white matter core as the intracerebellar nuclei, and the white matter is actually seen in the inner part of the folia as well as the innermost part of the cerebellum. Now we will see how these grey matter, the neurons, grey matter means the neurons of the cerebellum, how are they arranged and what are the different types of neurons you get. So we are now going to focus only at the cerebellar cortex. So when you see the cerebellar cortex, we can say that it is divided into 3 layers. The outermost is the molecular layer, then you have the middle Purkinje layer, and the inner part is the granular layer. These are the 3 different layers of the cerebellar cortex. And after this dotted line, it is the white matter, which is made up of nerve fibers alone. So let's see what are the what are the different types of neuron which we get in each layer of the cerebellar cortex. So first we will start with the molecular layer. So in the molecular layer, what are the cells which you can make out? So before that, I would like to say about the Purkinje layer, because the process of the Purkinje layer are also coming into the molecular layer. So it is better to define the Purkinje cells before we move on to the molecular layer. So middle layer is otherwise known as Purkinje layer because you get a single layer of Purkinje cells. So these red type cells are known as Purkinje cells, and the shape is just like a flask, isn't it? So you can see that from the bottom of the flask you have the axon, and from the top of the flask you have one single dendritic process which ramify in the molecular layer. One single dendritic process arises from the Purkinje cell, and when it reaches the molecular layer, it actually arborizes or branches. It gives off branches into the molecular layer. So dendritic process and the spines, the dendritic spines, that is how they explain the processes arising from the Purkinje cell into the molecular layer. Now what else you will get in the molecular layer? You will get one type of cell that is known as basket cell. So basket cells, we can see that it is actually lying horizontally. You can see the neuron, the cell body of the basket cell with the short dendritic processes, and the axon is actually running parallel to the folia but perpendicular to the dendritic processes of the Purkinje cell. And this axon will actually give collaterals which are in the form of a basket-like network, which will anastomose with the dendritic process of the Purkinje cell. Another type of cell which you get here is known as the stellate cell. Stellate cell are smaller compared to the basket cell, and they are actually seen towards the periphery, whereas the basket cells are seen towards the inner aspect of the molecular layer, whereas the stellate cells are seen towards the periphery, and they actually ramify, and their axons will give off branches which will anastomose with the or which will synapse with the dendritic spines of the Purkinje cell. So these are the two different types of cells which you get, that is basket cell and stellate cell. You have the dendritic spines of the Purkinje cell in the molecular layer. Apart from that, you have two more synaptic terminals, that is, one is the climbing fiber, which actually climbs up and makes synapse with the dendritic process of the Purkinje cell, and you also get the granule cells which are actually giving off axons. So if this is a granule cell, it is seen in the third layer, the granular layer, but it will give off the axon, and the axon will actually end as a T which are running parallel to the fibers of the basket cell, and these axon terminals will also make synaptic contact with the dendritic process of the Purkinje cell. So apart from these two neurons, you will get the dendritic process of the Purkinje cells, you will get the axon terminals of the granular cells, and also the climbing fibers synaptic terminal ending in the molecular layer. So you have to remember all these points. Now coming to the next layer, the Purkinje layer. Purkinje layer we have already mentioned. You have flask shaped cells with axon coming from the bottom, and you have the dendritic single process which actually arborize in the molecular layer. Now coming to the granular layer, the third layer, the granular layer, there you will get many granule cells, and along with that you will get large Golgi cells as well. So granule cells we have already mentioned, the cell bodies will be lying in the granular layer. The axons from the granule cells will be going up, and they will be actually branching as a T in the molecular layer, and they are known as, these T-shaped fibers arising from the granule cell are known as parallel fibers, and they will be synapsing with the dendritic process of the Purkinje cell, and their dendritic process will be actually making a synapse in the granule layer along with the axon of the Golgi and also the mossy fibers. So this is known as, this synapse which is seen in the granule layer is known as cerebellar glomerulus. So in the next session I will be talking about the white matter of cerebellum. There we will be mentioning more about the mossy fibers, the climbing fibers, how are they making synapse and all in detail. For the time being, you just know that in the granule layer you get the neurons of two cells, the cell bodies of two neurons, that is granule cell as well as Golgi cell. And you also have the synaptic meeting point, that is the cerebellar glomerulus. So we mentioned about the granule cell. Coming to the Golgi cell, they are a bit larger than the granule cell with dendritic processes moving on to the molecular layer. And we have the axon actually making the synapse with the cerebellar glomerulus. So it is the axon of the Golgi, it is the dendritic process of the granule cell, and the mossy fiber which are forming the cerebellar glomerulus. The dendritic process of the Golgi, they ascend up into the molecular layer and they synapse with the neurons, the processes of the neurons seen in the molecular layer. So when we talk about the neurons in the cerebellar cortex, once again we can just name the neurons. We have the stellate cells, basket cells in the molecular layer. Then you have the Purkinje cells in the Purkinje layer, and then you have the Golgi and granule cells in the granular layer. So we have altogether 5 types of neurons in the cerebellar cortex. When we talk about these 5 types of neurons, only the granule cells are excitatory, whereas the Golgi, the basket, the stellate, Purkinje all are inhibitory in function. Only the granule cells are excitatory. So in the next session, we will be talking about how these inhibitory response and excitatory response will be influencing the entire circuit when we discuss about the white matter. Now we are moving on to the intracerebellar nuclei, which are seated in the core of the white matter. Till now we discussed about the grey matter, the periphery of the cerebellum. Now we are moving on to the interior, where you will get the intracerebellar nuclei. So this is actually a cut section of the cerebellum, and we can see that if we consider this as the midline, on either side of the midline you have four sets of nuclei. So 4 pairs of nuclei on either side. So you have these 4 nuclei arranged from lateral to medial. So let's name them. The lateralmost, the biggest one, that is the dentate nucleus. Then you have the emboliform nucleus. Then you have the globose. Then you have the vestigial nucleus. So it is actually D, E, G and F. So that is the order, D, E, G and F. So that is how you have the intracerebellar nuclei arranged. So let's see one by one. So when we mentioned about the Purkinje cell, we mentioned about the axon of the Purkinje cell. That is actually the main output from the cerebellar cortex. The entire information from the cerebellar cortex comes out through the axons of the Purkinje cells. But the Purkinje cells which are seated in the floccular-nodular lobe, their axons won't be going to the intracerebellar nuclei, but they will be directly going to the vestibular nuclei. That you have to remember. So once again, the cerebellar cortex, the entire information from them will be coming out from the cerebellar cortex through the axons of the Purkinje cell. So axons of the Purkinje cell are the sole transmitters of information from the cerebellar cortex to the intracerebellar nuclei, from where it is taken outside the cerebellum. Okay, so all the axons of the Purkinje cells are meeting in the intracerebellar nuclei, except the Purkinje cells of the floccular-nodular lobe, where they directly go to the vestibular nuclei. That also you have to remember. So let's see one by one. First one is dentate nucleus, which actually belongs to neocerebellum, the highly evolved. So especially in primates and human, the dentate nucleus are very large. It is in the form of a crumbled bag-like pattern with the opening of the bag which is anteromedially, and this is considered as the hilum of the dentate nucleus. So the fibers, so inside the bag, inside the dentate nucleus, we have the fibers which are passing through the superior cerebellar peduncle, the efferent fibers. The fibers which are coming from the dentate nucleus will be passing through the superior cerebellar peduncle. And where do they go? They go to the red nucleus and thalamus, the ventrolateral nucleus of the thalamus, as dendatorubral and dendatothalamic fibers. Okay, so you have the lateralmost nucleus, the dentate nucleus, the large crumpled bag-like appearance which belongs to neocerebellum, the highly evolved. And the fibers, it is filled with fibers, efferent fibers which are coming out of the dentate nucleus, and they travel through the superior cerebellar peduncle to 2 main locations. One is red nucleus of midbrain as well as to the thalamus, the ventrolateral nucleus of the thalamus, as the dendatorubral and dendatothalamic fibers. Now coming to the nucleus medial to it, that is emboliform nucleus. It is actually seen at the hilum or just medial to the hilum of the dentate nucleus. And from this emboliform nucleus, you have the fibers again passing through the superior cerebellar peduncle. They go to the red nucleus of midbrain. And next to that, you have the globose nucleus, which is actually seated, which is a bit smaller than the emboliform. It is actually seated between the vestigia and the emboliform nucleus. So we mentioned about the emboliform and globose nucleus. Together you call it as nucleus interpositus, the nucleus which is positioned between the vestigial and dentate. That is what is meant by interpositus. So globose and emboliform together known as nucleus interpositus, and they actually belong to paleocerebellum. So they will be receiving fibers from the paleocerebellum. And the medialmost one is actually closer to the roof of the fourth ventricle near the vermis. And this is actually the vestigial nucleus, and this is actually belonging to the archicerebellum. So what will happen to the fibers coming out of the vestigial nucleus? They will be directly going to the vestibular and reticular formation, vestibular nuclei and reticular nuclei. Okay, so that is how the fibers emerge from the intracerebellar nuclei. So when we mention about the Purkinje cells, all the information, all the efferents from the cerebellar cortex will be coming from the Purkinje cell through its axon, and they will be meeting into the intracerebellar nuclei, except the one from the flocculonodular lobe, which will be going directly to the vestibular nucleus. And when we talk about the different intracerebellar nuclei from lateral to medial, we have the dentate, emboliform, globose, vestigia, D, E, G, F, and emboliform and globose position between the extremes, we call it as nucleus interpositus, and they belong to paleocerebellum, and dentate nucleus belong to neocerebellum, and vestigial nucleus belong to archicerebellum. So from the dentate it goes to the red nucleus and thalamus as dentatotubral and dentatothalamic fibers through the superior cerebellar peduncle. From the globose and emboliform, it goes to the red nucleus again through the superior cerebellar peduncle, and from the vestigia, which is archicerebellum, they go directly to the vestibular and reticular formation. Okay, so this is about the grey matter of the cerebellum in a nutshell. In the next session, I will be talking about the white matter, how these fibers ascend and descend into the cerebellum, how they make synapse, how they are excitatory in function, how they are inhibitory in function in detail. So keep watching, leave your comments if you find it useful. Please do share the video so that I will be motivated to do more and more sessions. Thank you, thanks for watching.